Hello guys. I will explain about prion disease. It is a disease that occurs due to the existence of two stable three-dimensional protein structures, can be transmitted between individuals of the same species, and in some cases, can even cross species barriers to transmit the disease. This table shows the prion diseases known in mammals. In humans, prion diseases are generally referred to as Creutzfeldt-Jakob disease. Additionally, a disease called Kuru, which was endemic in certain regions of New Guinea, is now known to have been a form of Creutzfeldt-Jakob disease. In other animals, prion diseases are known by different names. In sheep, it's called scrapie. In cattle, it's bovine spongiform encephalopathy, commonly known as mad cow disease, which caused worldwide panic. Prion diseases also occur in deer, mink, cats, and other animals. Although the symptoms of these diseases vary slightly depending on the species, it is now clear that they are all caused by the same protein called prion protein. I would like to first review some general concepts about protein folding into three-dimensional structures. This is commonly referred to as Anfinson's dogma, which posits that once the amino acid sequence of a protein is determined, it will fold into a three-dimensional structure with the lowest free energy through interactions between side chains, given ideal environmental conditions. It is believed that the amino acid sequence has been evolutionarily selected to form a structure with significantly lower energy compared to other possible structures. In prion diseases, the abnormally structured prion protein is denoted as AAA, PRPSC. It is now known that the disease is caused by the slow accumulation of this prion protein in cells, without being degraded. The SC designation originates from scrapie, a prion disease in sheep characterized by the animal's behavior of scraping against fences. The figure on the left shows the normal structure of the prion protein, labeled as PRPC. The C stands for cellular prion protein. The abnormally structured prion protein is formed when, for some reason, two alpha helices of the normal prion protein are converted into beta sheets, resulting in a structurally abnormal protein. The human prion protein is a relatively small protein, consisting of 253 amino acids with a molecular weight of 27,000 daltons. Prion proteins that have undergone structural abnormalities aggregate to form amyloid fibrils or amyloid plaques. The beta sheets of abnormal prions interact with each other, leading to aggregation and formation of protein clumps called amyloid plaques or amyloid fibrils. Since prion proteins are normally highly expressed in neuronal cells, the accumulation of abnormal proteins occurs prominently in brain and nervous system tissues. Once the accumulation of abnormal proteins begins, the process cannot be interrupted, and ultimately leads to the death of the individual. Endemic disease of Papua New Guinea, Kuru. I will explain the process of how the mechanism of prion disease development was elucidated. Although it is now recognized as a prion disease, it was discovered in the 1950s as a neurological endemic disease, a disease that occurs in specific regions or populations, affecting the 4 people of Papua New Guinea. This endemic disease was named Kuru. The boy in the photo has Kuru and cannot walk properly. At that time, it was recorded that there were 2,584 patients among the 4 people, of which 70% were adult women, very few 10% were adult men, and 20% were children. Among adults, there was a characteristic high ratio of women and few men. Today, we know that the cause of Kuru was due to the 4 people's custom of eating the flesh of humans, who had developed Kuru, primarily by women, for religious reasons. Research was initiated to investigate the mechanism of Kuru disease onset. As cases of mice exhibiting symptoms similar to Kuru disease were discovered, experiments were primarily conducted using mice. Brain tissue was extracted from mice with Kuru disease, and the cells were crushed and separated into components, such as proteins, nucleic acids, and lipids. These components were then administered to healthy mice. As a result, it was experimentally determined that after a long incubation period, mice developed Kuru disease, albeit at a low frequency. Through such experiments, in 1982, Stanley Prusiner proposed that Kuru disease is an infectious disease caused by a specific protein. He named this specific protein Prion. This experiment captures the key points about the research into Kuru disease. Subsequent research revealed that the gene encoding the prion protein, called the PRNP gene, is located on chromosome 2 in humans. However, in mice, it was found that the sequence of the prion gene in individuals that developed prion disease was identical to that of normal individuals, indicating that DNA sequence mutations are not the cause of prion disease. Furthermore, it was demonstrated that knockout mice lacking the prion gene do not develop prion disease, even when abnormal prion protein is directly introduced into their brains or administered in their feed. Researchers were greatly perplexed by such experimental results. The fact that a pure protein without genetic material like DNA or RNA could be the cause of prion disease, act as a source of infection, and spread the infection to other individuals was difficult to explain with the molecular biology knowledge of the time. For example, if someone were to die from eating a poisonous pufferfish, it's unlikely that another person would die from eating part of the deceased person's body. Even if they did die, it's inconceivable that a chain reaction would occur where someone else would die from eating the person who died from eating the first victim. This is simply because protein toxins would be diluted as they pass from human to human. If the cause were a replicating factor with genes, like bacterial or viral infections, it could spread infection indefinitely from individual to individual without dilution, as it would replicate within newly infected individuals. So, researchers couldn't conceive of a mechanism by which a pure protein could cause infection continuously over a long period, spreading from individual to individual. The possible mechanism has been discussed and hypothesized by researchers from various fields. The proposed model suggests that the misfolded prion protein acts as a seed, interacting with existing normal prion proteins and altering their three-dimensional structure to become misfolded. According to this model, normal prions with a proper structure are continuously supplied from normal genes, so if a single molecule of misfolded prion is introduced into the body, it will gradually increase in number and accumulate within cells that are producing the normal proteins. Generally, abnormal proteins are tagged with a short peptide chain of ubiquitin and degraded in the proteasome. However, it has been discovered that abnormal prions bind to the proteasome and inhibit its function. Consequently, when abnormal prions are present, other abnormal proteins also become difficult to degrade in the proteasome. As a result, AAA, abnormal proteins accumulate increasingly within cells, eventually leading to symptoms such as difficulty walking. This is the mechanism of prion disease onset. The incubation period is long, considered to be at least 8 to 10 years. Moreover, the fatality rate after onset is 100%, with death occurring about 1 year after symptoms appear. Global epidemic of bovine prion disease, mad cow disease, BSE. Here's a brief explanation of bovine spongiform encephalopathy, called BSE for short. It is also commonly known as mad cow disease. BSE was first reported in the United Kingdom in 1986. The disease got its official name bovine spongiform encephalopathy because the brains of affected cattle had a sponge-like appearance. As shown in this image, cattle infected with BSE display neurological abnormalities and are unable to walk normally. The BSE epidemic began on British farms in the late 1980s and early 1990s, eventually spreading throughout Europe and even reaching North America. The peak of the epidemic was in 1992, when approximately 37,000 cattle worldwide were found to be infected. Between late April 1996 and the end of 2001, about 5.3 million cattle were culled globally. How BSE spread rapidly worldwide. The rapid spread of BSE worldwide can be attributed to the following process. In every country, meat from cattle infected with BSE cannot be used for human consumption. As a result, these infected cows were processed into meat and bone meal, which was then used as a raw material for cattle feed. When healthy cattle consumed meat and bone meal from BSE-infected cows, the abnormal prion proteins were ingested by the healthy cattle. This triggered the accumulation of abnormal prions in previously healthy cows, following the mechanism described earlier. Complicating matters further, abnormal prion proteins are difficult to digest in the stomach and can retain their properties even when heated. These characteristics of prions derived from BSE-infected cattle in meat and bone meal contributed to the spread of the disease. It is believed that, when ingested orally, the abnormal prion proteins, without being digested, enter through the lymphoid tissue in the intestines, and then invade the brain via nearby nerve fibers. The proliferation of cows with BSE is problematic in itself, but the situation became even more serious when it was discovered that BSE could be transmitted from cattle to humans. Unfortunately, the three-dimensional structure of bovine prion protein is very similar to that of human. It has been found that consuming meat from BSE-infected cattle, especially meat near the spinal cord and brain where prion protein accumulation is significant, can lead to the ingestion of altered bovine prion proteins into the human body. Additionally, abnormal prion proteins are highly resistant to heat, making it difficult to inactivate them through normal heating processes. To inactivate them, heating at 134 degrees Celsius for 18 minutes is necessary. Three-dimensional structure of the abnormal prions is so stable. These abnormal prions can then transform normal human prions into their pathogenic form. The discovery of this cross-species transmission from cattle to humans has greatly made worse the issue. As these newspaper articles indicate, mad cow disease shook the world. However, after establishing rules prohibiting the use of infected cattle as raw material for meat and bone meal, the outbreak of mad cow disease eventually subsided worldwide. While the BSE epidemic in cattle has been largely controlled, sporadic cases of Creutzfeldt-Jakob disease continue to occur naturally in human populations at a low rate. It affects approximately 1 individual per million per year worldwide, with about 350 cases diagnosed annually in the United States. The disease is invariably fatal, with most patients dying within a year of onset. A healthy person who receives a corneal transplant from a patient with naturally occurring Creutzfeldt-Jakob disease may also develop the disease. Unfortunately, there is currently no cure for Creutzfeldt-Jakob disease, and it remains an incurable condition. Researchers are hoping to develop treatments such as vaccines that stimulate the production of antibodies that bind only to abnormal prion proteins. Prion-like proteins that also exist in yeast. In recent years, prion proteins have come to be understood in a broader sense. They are now widely regarded as proteins that have a catalytic effect, acting on normal proteins to alter their three-dimensional structure and transform them into a different stable conformation. In this broader definition, prions do not necessarily need to share homology with the prion proteins that cause prion diseases in humans and cattle. Under this expanded definition, the yeast Saccharomyces cerevisiae contains more than 10 types of prion proteins, including SUP35. This means that in yeast, at least 10 types of proteins have been found that can act on normal proteins, convert their structure to the same abnormal form as themselves, and accumulate within cells. Moreover, in the case of yeast, the accumulation of abnormal proteins does not seem to be necessarily detrimental to the cell. In principle, a protein's three-dimensional structure is overwhelmingly stable in only one conformation compared to others, which is essential for maintaining an orderly metabolic system in living organisms. However, it has become known that in certain yeast growth environments, the accumulation of such abnormal proteins may actually be advantageous for survival. Similar proteins might also be found in higher organisms.
はい。ではこれで終了します。
If you have not found it yet, keep looking.